Hello, my name is Sadie Shelton, and today is March 27th, 2021, and I am going to be talking about anatomy and enzymology of fat absorption today. So first, we are going to start with a little bit anatomy of the different components that help with fat digestion. So the most important thing is the liver here. It stimulates and has a bunch of different functions, but what's really important is that next to the liver is this little pouch called the gallbladder. So the gallbladder releases bile, which is really important in the preparation of the digestion of fats. So in the gallbladder, there's a substance called bile. And the release of bile is due to a bunch of different hormones, which I'm going to talk about now. So it all starts with parasympathetic stimulation from the vagal nerve or the vagus nerve, causes the gallbladder to contract. So there's a couple other different hormones that also help release bile. So when the stomach is digesting food and the acidic chyme is sensed in the small intestine, the presence of acid in the duodenum causes the release of secretin. Secretin also stimulates liver parenchymal secretions such as bile. Also, when we sense fats in the duodenum, this causes the release of another hormone called cholecystokinin, which is released by the pancreas. It's also called, CCK, releases the sphincter of Oddi. So the sphincter of Oddi helps release the bile actually into the small intestine. So bile is produced by the liver and is stored in the gallbladder. When it is stimulated by the vagus nerve to contract and release, it travels down the common bile duct into where it meets the sphincter of Oddi. The sphincter of Oddi is kind of the gatekeeper and helps either allows or doesn't allow various hepatic, gallbladder, or pancreatic secretions, enzymes, hormones from entering the duodenum. So once this CCK relaxes the sphincter of Oddi, the bile is then released into the duodenum and then can help with fat digestion. So next slide, we're going to talk about a little bit about fats, so the chemistry of a fat. So this right here, up in this top left corner, is a triglyceride. So as we can tell that it has a carbon backbone, and then what are these little tails are 3 fatty acids. So as we look at the fat, there is a ton of carbons. So all these carbons make this triglyceride extremely hydrophobic and extremely lipophilic. So it loves fats, of course, since it is a fat, and hates water. So this causes problems because when you're in the duodenum or in the small intestine, there's a lot of different secretions and waters and stuff like that. And so when we go to digest it, it's not in a prime. This formation or structure it's in right now is not the most ideal. So if we look over in this second picture in the bottom right-hand corner, we can see that bile can coat the lipids. Bile is what we call amphipathic, so it has a hydrophobic side and a hydrophilic side. So one side hates water and one side loves water. So what happens is this bile will coat the fats, and then you have the hydrophobic side is going to mingle with the lipids, so this kind of inner side. So it coats it so then it can form these little balls. The hydrophilic side associates with the water and faces outward so it can travel in the small intestine easier. So as we can see, fats are extremely hydrophobic, and bile helps make it a little bit more hydrophilic by covering it. So our next slide, we're going to talk about bile a little bit. So as I already said, bile is made in the liver and it's stored in the gallbladder. It is made up of different components. It helps with digestion, absorption of fats by emulsifying and solubilizing the fats into breaking it down, making it more hydrophilic, because as we mentioned earlier, it's extremely hydrophobic, and also excretion of different waste products from the liver. So bile is made up of a bunch of different stuff. Its main composition is bile salts, and that is what actually helps break down the fats. Bile salts they travel through the duodenum and then we reuse them. So the body doesn't have an unlimited store of bile salts. We reuse what we have over and over and over again. Once it reaches the ileum, it is then reabsorbed into the bloodstream and transported back to the liver so it can start the process of breaking down more fats. Sometimes the bile salts can reach all the way to the colon, and then when that happens, colonic bacteria will break down the bile salts to their precursors called bile acids, and that is redistributed through the blood back to the liver so we can use it again for fat digestion. Some more waste products are phospholipids and cholesterol. Cholesterol is excreted in the feces. A little bit of it is reabsorbed, but most of it is excreted in the feces. And then we also have bile pigments, which is also known as bilirubin, which is the waste product of broken down red blood cells. So that is also excreted in the feces. So that is kind of what bile is made up of for fat digestion and absorption. Mostly we're going to focus on the bile salts. Okay, so the next picture, we're just going to talk about just fat digestion. So breaking down this triglyceride, which we talked about earlier, the carbon backbone with the 3 fatty acids, into more of like a monomer. So a monoglyceride, one part of the triglyceride, so it's smaller, more broken down, easier to be reabsorbed. So as I mentioned earlier, fatty acids are not fatty acids, but triglycerides are extremely hydrophobic, so they will meet up with other triglycerides and form these large fat droplets. These large fat droplets don't have a lot of surface area that is hydrophilic, and so it's really hard for the bile salts and other enzymes to help break down those fat droplets. So we have these large fat droplets, they accumulate, and there's not any surface area, there's nothing for them to connect to. So what happens is bile salts from the bile is excreted from the gallbladder into the duodenum, and they meet up, and it's called an emulsion. So then, as I mentioned in that earlier slide, you have the hydrophilic is on the outside of the bile salt, and the hydrophobic is on the inside to meet up with the fat, and you take these large fat droplets and you make them into something really small. So this picture on the top right is what an emulsion looks like. So you have your bile salts, you have, this is an emulsion or a missile. So this is kind of like what it looks like. So you have the bile salts, and then you have your different components of the fats are inside this emulsion. You have your diglyceride, your monoglycerides, phospholipids, just all the different types of fats. So the fats are going to congregate in the middle. The bile salts, because they are hydrophobic and hydrophilic, are going to face the outside. So when the bile salts come in, it creates a bigger surface area for these enzymes to come in. And so that's what happens with the micelles. So micelles are also kind of what this picture shows. It increases the surface area from the bile salts and allows enzymes to come in. So you have 2 enzymes that come in. You have lipase and you have colipase. So ACE is the breakdown of something, and of course lipid is a fat. So these enzymes are going to break down fat. So what they do is, as in this bottom right picture, they take triglycerides and then they break them down into monoglycerides, and then even further, the colipase breaks them down into fatty acids. So once we have these free fatty acids, this is what's going to be reabsorbed into the small intestine. It's finally been broken down small enough that it can be reabsorbed, which is what we're going to talk about on the next slide. So on this last slide, we're going to talk about the fat absorption. So we've taken these really large fat droplets that are really difficult to break down. We've broken them down into smaller droplets with the help of the bile salts, and then we've increased the surface area and allowed the lipases to come in and break down the triglycerides into the fatty acids. As mentioned earlier, we also have cholesterol, which is this oval. Cholesterol is secreted in bile. It sometimes comes from other sources, but it also is a fat, so it can sometimes be reabsorbed. So here, with this 3A, we have our free fatty acids. Sometimes monoglycerides, they are small enough, they will be reabsorbed. They are reabsorbed through facilitated diffusion. So that means that there's a high concentration on the outside of the cell of the small intestine, and it's going to be able to, because it's lipophilic, it can pass through it can pass through the membrane without any ATP or anything, because A, it is following the high to low concentration gradient. B, it is lipophilic. And so the lipids are going to be able to pass through the membrane without meeting any resistance. So the fatty acids and monoglycerides are going to pass through the membrane with facilitated diffusion. Cholesterol is a bit bigger, it's a bit chunkier. That needs a transporter. So transporter is going to meet up It's going to help transport that through the membrane of the small intestine. Next, we have the fatty acids are going to go through the smooth endoplasmic reticulum, which is going to revert them back to triglycerides. So we had to break it down so it could pass through the membrane. It's been broken down, and so now it's going to need to turn back into triglyceride because that is what we're going to reabsorb. So these fatty acids and monoglycerides pass through the smooth ER, meet up with the cholesterol, and then combines with cholesterol. We have cholesterol, triglycerides, and protein, and it forms what's called a chylomicron. So it's just a little package of fats that is ready to be redistributed to the body to be used. That will then pass through the Golgi apparatus, which is the FedEx of the cell. It gets packaged up and then does cytosis or exocytosis to exit the cell. So usually what happens with reabsorption is it's reabsorbed in the capillary and then distributed in the circulation system. So these chylomicrons, they're too big, they're too hydrophobic to just be easily reabsorbed into the capillary. And so what's going to happen is it actually is going to be reabsorbed through the lacteal, which is a part of the lymphatic system. It's like a little duct, a little kind of like just a little extension, like a villi kind of type thing, but it's called lacteal, and it's for the lymphatic system. It reabsorbs lymph, and it's going to reabsorb the chylomicrons, and then the lymph is going to, the chylomicron is going to travel in the lymph all the way to the vena cava, where then it will be re-met up with the blood. And that is how fats are digested and reabsorbed.